So ethnomycology, it's basically just understanding the relationship between, well, it's just this ongoing study between humans and fungi and where they intersect. And the best way to think about it is utilizing fungi for food, medicine, and technology. Get a little bit of a background, some ethnomycology examples through time, medicinal and functional background, some chemistry, psychedelic, spiritual use, ecological relationships, historical and contemporary context, and then future prospects. So backgrounds in evolutionary biology, I like to start from the beginning of time. From the beginning of life, basically. So has anybody heard of a phylogenetic tree of life? So it's really interesting, but you can take DNA from anything, and then you can use molecular clock dating through mutational shifts to see how closely related they are over time. So this is, you see there's a central point right here. It's really hard to see. And then it evolved all multicellular complex life. These are all the different domains of life. We'll simplify this a little bit. So this is grossly oversimplified, but it's the same concept. And this is specifically a phylogenetic breakdown. So all these little tuning forks is phylogeny. So where you see the very bottom there, LUCA, it's an acronym for your greatest grandmother of all time. It's the last universal common ancestor. And then where it tuning forks, that's a divergence in species. So what defines a new species? Many different things, but mainly that they were once one, and now they can no longer reproduce and make viable offspring that is fertile. Because you can get into donkeys and mules and all that kind of stuff. We won't. But basically, that can happen for all sorts of reasons. A really good example, Gondwanaland, anybody? So Pangea, you know, when the Earth was one giant landmass and then started floating away from itself, that's that space and time, and because of epigenetic shifts or genetic drift, it can be random, it can be highly selective, but populations will change on a population scale over many, many years. And they change so much that if you brought them back together, they cannot mate anymore. That is that tuning fork. Now, if you follow the tuning fork all the way out to the right, you see eukaryotics. That's nuclei-bearing organisms. We're nuclei-bearing. So are fungi, so are plants. And we shared a common ancestor, and a lot of this stuff changes over time. When I graduated, my understanding of the common ancestry between humans and fungi, we're closer related to fungi than we are to plants, which is really important, and consider that fungi fight the same things that we fight. But let's not get an anthropocentric view on this. There wasn't a coevolution because of us. Come on, egos. These work for animals. They work for insects probably. They work for all sorts of things that are closely related. But they fight the same viruses, the same bacteria, the same cancers, you know, certain things like that. And fungi are the grand chemists of nature. They're like a janitor with an infinite key ring, and given enough time and resources can unlock any door. It's pretty cool. So they create really unique chemistry through biosynthesis. They organically can create really complex molecular structures, enzymes or sugars or whatever, that can fight really cool things for immune support, or we can go into the functionality in a minute. But if we took that and zoomed in a little bit, you can see animals are on the bottom, and then these are all the different genus of fungi. Most of the fungi that we're all familiar with or understand or look at or can visibly see as a fruiting body are pretty much basidiomycetes. There are some ascomycetes like cordyceps or morels, and that generally is how they produce and mate, is how they're differentiated, but mostly everything that you will ever encounter is going to be a basidiomycete. And in this evolutionary historical context through time, the oldest accepted evidence for fungi life was fossilized spores in mycelium that date to about 1 billion years ago. We separated from fungi, started 650 million years ago, but now it's 1.1 billion years ago. So that had to evolve to that point. So arguably the oldest organisms on planet Earth, probably fungi. It really brings credence, in my opinion, to panspermia, so look into it. Being seeded from another universe, basically, spores floating through the space vacuum. There is evidence for fossilized mycelium in lava tubes. It dates about 2.4 billion years ago. It's half the age of planet Earth. It's pretty cool, but it's highly contested. Who knows? And this is just a visual. This is just a visual kind of map of time looking at these different points in fungal evolution. Oldest animal fossils, we have fungi way down there. And then about 55 million years ago, we have a symbiotic relationship. I like to go outside of animals, or outside of humans in relation to fungi as well. So I'll talk about other aspects of ethnomycology not related to humans. But ants from the genus Atene, leafcutter ants. Anybody been to South America? So seeing leafcutter ants kind of just on their trail bringing leaves. They take about 15 to 20% of the leafy greens out of the Amazon. It's a big number. And they bring them, these chunks of leaves, to their underground, I always say this and never know what it is, but an underground layer, like their ant cave system, whatever you want to call it. And then they chew it up and they feed it to a mycelium. And that mycelium in turn will feed them sugars, probiotics, whatever the colony needs to survive. And it's a true symbiotic relationship where they would not survive without one another. And it dates back about 55 million years.
So they're feeding them to the mushrooms?
The mycelium. Yeah.
To the fungi.
So the mycelium is coating the walls of their underground layer. And they'll kind of go to it and feed it, and it'll give them food in return. So it's this symbiotic relationship.
It sounds like almost like the way a compost pile works.
Yeah, no, I was thinking compost.
Like like you feed organisms in your compost pile, and then the compost pile gives you beneficial bacteria for your garden.
Yeah, and fungi are true ecosystem engineers, and they can control not only the life you see around you from start to finish. They'll take life, but they can control the microbial levels. So they are true ecosystem engineers. Like, it's what we see here as a success story is because of fungi that we don't see. And when a queen is born, they'll take a little bit of that mycelium, they'll tuck it in her exoskeleton, she'll go out and start a new sort of brood, if you will, or a new population, and then she will inoculate that new underground layer with a copy clone of the mycelium. So there's just little patches of cloned mycelium all throughout the Amazon underground. Trophic cascading, pretty cool concept. Classic example is wolves, Yellowstone, biodiversity. I guess anybody know that example? Real quick, we're going to that one, or else it doesn't work. So when you bring wolves back into Yellowstone, it increases biodiversity. And they're like, how is this going? What is going on here? And it's because it's not a direct effect. So because of their presence along river waterways, the ungulates are more skittish and less likely to eat the trees or brush seedlings. And then those will grow up, which allows beavers to have more food. The trees will grow up, which allows birds to be there. Birds will spread. It just keeps building on top of itself from a keystone species like wolves. So bees in trees and bears and mushrooms, they're also very related. It turns out that bees benefit significantly from scratching the mycelium of certain mushrooms that are on trees, the mycelium, and then they'll drink the fluids that it exudes. And it gives them probiotics, and it helps them fight things like varroa mite disease, colony collapse disorder, stuff like that. But what complex interaction that's going on is bears will go through, for various reasons, will scratch trees to make noise, or it's a mating thing, or whatever. And that scratch will allow polypore, like they're called polyporish mushrooms because they have tons of pores on the bottom, but branch fungi that are found on the sides of trees. It'll allow spores to land there and then grow, which in effect will increase the health of the bee populations that are native to the area. But there's the example, I guess, is just mainly that there's a complex interaction between fungi and our environments. So that's ongoing. I don't know the age of when that kind of started, but then we have getting to the human examples. 20,000 years ago, 18,000 years ago, there was a past 10 years they found a cave in Spain. Cantabria, El Moran Cave, I think is what it's called. And they called her the Red Lady. She was adorned in red ochre. It was a ceremonial burial. But why she's interesting to the mycological community is because they found spores in her teeth. They found Amanita genus, so they assume it was a psychedelic ritual burial. And they found boletes spores in her teeth. So 20,000 years ago, there was evidence for medicinal use as well as for sustenance for food, basically. Then you have 5,000 years ago, I think it was just like 2000s, whatever, people are hiking in the Alps. I call it the Austrian Alps. I'm partial to Austria. Kind of combines multiple regions, but they're hiking up there. It's glacier territory. They found a body half out of the ice, and they're like, oh no, called Mountain Rescue. Mountain Rescue's like, this is a mummy. This is not from any period of time where I've been alive. So Ötzi, the Iceman, dates to about 5,000, 6,000 years ago. He was well preserved. Glacier drift moved away and exposed his body. Why he's interested? There's a ton of articles on Ötzi, podcast, stuff like that. But why he's interested to the mycological side is they found fungi on him, which is really interesting. He had it worn as jewelry or tools, and he had a medicine pouch. The tools, the tinder conch is a really good example. When it's cored out, you can put an ember in it, and it can keep an ember burning as long as it doesn't get wet for days, if not weeks, and they can use it to seed fires. So as nomadics were moving in colder regions or moving from spot to spot every day, they could bring the fire with them. He was wearing a birch polypore, which was arguably used to treat internal parasites. So, they had a really good understanding of medicinal use as well as tools. And there's a couple other examples. And then, on our timeline, I don't know, 4,000 BC, maybe 3,000 to 1,000-ish. You have the Egyptians. There's a ton of information on Egyptians, not only cultivating psilocybe mushrooms on grain, but it was written in a lot of their petroglyphic historical contexts that they were reserved for nobility, just like in Asian continental areas, same idea. If you were caught collecting mushrooms, you should be giving it to nobility or the pharaoh. Otherwise, you would be executed. It was not for commoners, apparently. And then that was for sustenance. And then for psychedelic usage, it was food of the gods. It was immortality. It was well known that fungi played a massive role, whether it was spiritual use or for food, even in that time. And even in their construction of buildings, you have like psilocybe type structures that were really important to the communities. And then there were a tablet prohibiting commoners from eating mushrooms. I mean, there's a ton of evidence for that kind of stuff. And then you have Mesoamericans. A lot of this comes from ritualistic type uses in a spirituality context using psychedelics. There's many psychedelic mushrooms that are found in Mexico and Central America, and more are being discovered on a regular basis. And there's tons of really cool evidence for these ritualistic uses of these mushroom stones. They were probably like designating a safe space for people to come to have these experiences. If they weren't well known or you didn't know where to find them, you would find these stones, and they would designate areas of where these rituals would take place. And this is a list of all the, since 2006, it's bigger now, of the hallucinogenic considered species through Mesoamerican, through historical context of what they consumed. There's question marks on a lot of it, because they're like, there was one story of they would use a mushroom as a stimulant, and no one's been able to figure out what that mushroom is, so there's a lot of really cool chemistry that needs to be figured out. This is just a map of the world distribution of hallucinogenic species of psilocybe. Each dot is one or several localities, and they're mainly found in populations of people. Same thing with not only functional and food mushrooms, they're mainly found in disturbed forests where people are, and there's this co-ecological evolution going on there too. And then you have the Greeks on our timeline. What's this, like 1,000 BCE or something like that? I don't remember, up to 400 or something. The Eleusinian Mysteries. If you're interested in a 1,000-year uninterrupted underground psychedelic ritual, read The Immortality Key by Brian Morescu. Great modern text. He didn't discover all of this stuff. Carl Ruck did, and that was like 60s, 70s, and he was laughed out of the department because like, people didn't use psychedelics, and it turns out there's a lot of really, really concrete evidence of all of this stuff. Yeah.
Can you repeat that book name?
The Immortality Key. Immortality Key. Brian Morescu. Don't ask me how to spell that. But then it's in the mythology too. Persephone, Demeter going to the underworld. You think about what is one holiday that pretty much the world celebrates on the same day every year. Sorry? It's close. Did you say Christmas?
Yeah.
It's close to Christmas. It's like 4 days away from Christmas. New Year's. Think about it. What do we do for New Year's? We think back on our year. We set intentions for the new year. And we try to do better and maybe stop drinking so much. These are all psychedelic sort of processes. And it's probably where that entire thing was birthed out of, honestly. The psychedelic brew that was consumed is highly contested. It's been proposed by Wasson in the 50s as Amanita muscaria, but then have you ever tried Amanita muscaria? The way they write about it? I mean, I wouldn't say it's glorious. So maybe it was a phenotypical evolutionary shift in its chemistry that changed over time. But that mushroom doesn't make a lot of sense. The most argued ones that probably do are a psilocybe-containing mushroom, because you can see here they're clearly giving each other mushrooms. But here depicts, it's written there, Demeter accepting an offering of wheat as part of the rites of the Eleusinian mysteries. And the brew was called the kykeon, and it had 3 ingredients: wheat, water, wheat flour, water, and mint. So there's a couple things going on there. I believe, or my opinion, based on what I've read, is it's ergot containing LSA compounds. LSA is a precursor for LSD, and they probably brewed it as sort of a wine, and they tried to figure out the chemistry in a modern sense. If they had like wood ash, they had some wine, they had some ergot, and they fermented it over a course of time, they could probably remove the noxious toxic compounds and have something close to an LSD type wine. That's the coolest one I've heard. I think it's pretty cool. And handing wheat makes a lot of sense because ergot is a fungus that infects wheat. But mint, that's interesting. As you may know, in South America, an interesting psychedelic plant that is in the mint family, Salvia divinorum. Completely different continents, so who knows? But mint is interesting there. But Salvia is in the mint family. The Greeks also knew a bit about cultivation, which is really cool. They knew if they dug out, I think it was fig tree roots, and they buried manure, they could get piopino mushrooms, and people had figured out how to like grow that naturally in their environments, which is pretty cool. You have a lot of historical writing about medicinal use of fungi as well, which is pretty cool to see, but it was kind of split. In the Greeks, it was either poisonous or it was good for you, but you just had to know what to do in that sort of situation to not die or get sick. And then everyone really talks about sort of this coevolution and epigenetic evolution of taking doses of psilocybin mushrooms for many thousands of years and having rapid cognitive functionality or brain growth even. So it's called the stoned ape hypothesis at this point, but it has to do with arboreal primates getting into the savanna and finding ungulates and finding psilocybe mushrooms going on the manure, and then having a population that starts consuming these mushrooms and having better ability to survive in their environments, and you do that over generations, you could have a rapid growth in consciousness and awareness, problem solving, leadership, all the things that we need today through psychedelic usage. And then I like to focus a little bit more on as native as I can on a context of the use of food, medicine, technology by native people. The best accounts that I could find are in California. Unfortunately, a lot of the literature is lacking for the historical use of fungi in most cultures, honestly, if it wasn't already written about. So like Native Americans in South America, you had these anthropologists coming through in the 1900s, throughout the 19-whatever. And when they would write the accounts, they would write in nauseam about the plants. And they would take those plants back and they'd run chemistry on them and understand them. But they would just write fungi or mushrooms. So that'd be like an anthropologist going into a community and saying they ate animals and plants. Done. You're like, okay. So it's just, unfortunately, the historical note-taking of the use of mycology is just underrepresented and not really understood. So unfortunately, a lot of that is gone. So we have California Native Americans used a lot of fungi, it turns out, and they're understanding a lot of different complexities here. So this is just a underrepresented map, probably, of the tribes of California. This is all of the documented information they can find on mushrooms used as food, technology, or medicine. They used about a lot.
Can I ask you a
Yeah.
question? What I think you touched on this a little bit, but what do you mean when you say mushrooms uses technology?
I'll get to that.
Oh, thank you.
Yeah. They have computers. No, I'm just kidding. And this is underrepresented as well, but out of all those lists that I just kind of burned through, they were able to identify about 36 unique species in California. I can barely name 36 species. That's pretty crazy. 19 fungal families. 2 tribes were documented to use truffles. Yeah, mushrooms poorly represented. Descriptions, I went over that. For medicines, right? So I kind of copy pasted these quotes from the papers I was reading because they're that cool. But they did have an understanding, maybe not to the level that we have today, for the use of fungi for medicines, for immunomodulation, for blood pressure, for viral infections or cancer or whatever. But their version of that would be understanding that if they had a person that was sick or elderly and sick or whatever, they knew how to make broths, and they would specifically hold mushrooms for the sick individuals to feed them to help with their immune defense in that process. Example for the wood ear, which is pretty interesting. They would boil it and drink the broth, but there was one account where someone had blood poisoning, and they soaked their leg in it, and they were able to get benefits as a topical, which is really interesting. Fungi in topical use is something that's getting more excitement around today for skincare products using like Ganoderma or reishi mushrooms. It's really interesting. Puffball spores, they would use the spores to stop runny noses. They just shove them right up in their noses, which is pretty cool. And there was one account where a Native American woman got shot in the face with an arrow through the mouth. They pulled it out and they packed it with puffball spores, and they were able to stop the bleeding, which is really interesting. And they would use a decoction of the red-edged yellow tree mushroom. They had their own name for it. It was probably chicken of the woods. And they would drink it as a cathartic to cure rheumatism. And then something called poultice. I didn't look that up, so I left it out. I was like, what's poultice? Technological uses. So they had pretty simple ones, but like we mentioned, like the tinder conch, they used a mushroom called a punk. And they would core it out, and they would keep embers alive multiple days at a time. And then they would use the red-belted conch. There's many different red-belted conchs. Like this is sort of a red-belted conch. And they would use the bottom of it, this abrasive side, which is the spores. They would use it to polish buckskin, which is really interesting, just by rubbing the mushroom on it. And then, I don't know what this mushroom is. It's a gnarl, like have you seen trees with like these crazy gnarl growths? So they would use those as housewares. So like cups and bowls, they would burn it and core it out, and they would have utensils basically they would use for eating or drinking. And then there was one account of using puffballs are really interesting. So if you find a puffball, some are edible, but some, when they get a little bit beyond edibility, they start producing a ton of spores on the inside. You ever seen someone like squeeze one and like, oh yeah. When it gets to that point, the outside's very leathery, and you can peel it off. And they would use that for various things. And one of them was like a maraca, basically, or like a baby toy, something like that, which is pretty cool. By putting rocks in it and tying it up on a stick. Food. There's tons of ways to consume fungi. What they like to do a lot of times was they'll make broths out of it. They will dehydrate the mushrooms a lot of times for long-term storage. They will this kind of goes into what they also eat. Were chanterelles, lion's mane, which I thought was pretty cool. black trumpet, hedgehogs, oyster mushrooms for sure. But what I thought was really interesting, a couple different points on when they were eating them, was thinking about the future and their harvesting techniques. That's what I thought was really cool, this interconnectivity between the ecological future of their environment and they were able to think ahead, basically, and not over-harvest or unsustainably harvest, and help spread spores in that process. They would use very specific harvesting techniques when they would find an area where these fungi would grow, whether it was like matsutake or chanterelles or boletes or whatever, and they would document it and they would consider it a garden. And they knew to come back every year, and they knew that they would take the older specimens. They would do a couple different things. They would leave the babies on purpose. They would specifically leave some for other animals. That's a really big Native American thing, where when they would grow crops, a lot of times they would leave some there for other animals or to give back to the earth. And that's what we do today, even the 30% rule. You want to leave about 25 to 30% of what you find, so those spores can drop and it can repopulate every year. They knew about mycelium, it turns out. They knew not to disturb the mycelium. If they did, they would cover it back up to protect it. Yeah, there's a lot of cool examples on that one. For the oldest specimens, they would a lot of times leave because they'd be buggy or whatever, but they knew they were dropping spores, so they would take them back and they would purposefully try to spread the spores around their encampments, basically, to spread spores. Spore dispersal. Typical camp was considered to be surrounded by bushes, heavily weighted down by drying fungi, which is really cool. It's this complex interaction between where they would be one year and they would be nomadic, right? And they would leave all of their mushrooms to dry around their camp. So when they come back the next year, they would probably have mushrooms growing in those areas. And the ecological relationships understood by Native Americans is just mind-blowing. They knew about seasonality, obviously. They knew the complex interactions between what's called mycorrhizal species or ectomycorrhizal species in the specific trees that they grew next to, or bushes or shrubs. They knew they knew elevation. They knew at certain elevations, certain mushrooms would grow. They would have to find the right tree, and they would know all of those complex interactions. These are the things that we use today to go out and forage our own mushrooms. One thing that I thought was pretty cool is that they practiced to increase mushroom diversity and mushroom dispersal and growth was by purposefully burning the areas, which for various reasons, there's a term for it and it escapes me right now, but some fungi like the heat. For some reason, like burn morels is a good example. A fire comes through and something either with the chemistry of the ash that changes the pH or the heat that's applied to the mycelium layer at that point will trigger the mushrooms to grow in high amounts. But then they're looking at this today as a technique and tool for future, for like going back to how it used to be, because our current for fire mitigation is mind-blowing. We'll go into that in a micromediation sense, but does anybody know, I'll jump ahead a little bit because it's interesting, the most technologically advanced technique for dealing with fire mitigation right now? Yeah. Burning it. But the worst part is they will pack in and pack out the underbrush. So they'll go in off the grid areas, grab it, and take it out and burn it. Like that's where we're at today. Doesn't make a whole lot of sense to me because there's a lot of cooler ways to approach detritus material, and we'll get into that. Yeah, so next topic that we're kind of going to dive into just a little bit is the chemistry of fungi. Is there any questions about the couple examples that I gave? Yeah.
So I think you'll get to it. I live in Northern California, and we just had a regional park that was given back to the native tribe. What's left, sadly. But is there anything like coming out in research where they're going back and revisiting like mushrooms that can now grow because they're replenishing the earth and they're going back to fire preservation or fire mitigation?
There might be, hopefully, I don't know, but there is a cool project that I'll talk about called Fundus, which is the Fungal Diversity Survey, and Gabriella runs that. She's super cool. They're a really cool project, and they're mapping out fungi and getting a good understanding of the diversity, the biodiversity of fungi all over, because that's where we're starting from, to understand what's even out there right now. And seeing how that shifts over time is now starting. Fungi, interesting organisms. Grand chemists of nature, I said that. Biosynthesis. So they're able to produce through many different processes, through a genetic level, and taking their environment and changing its structure into different forms. So fungi are a lot like people in that they breathe in O2, they breathe out CO2, they eat. It turns out we have an internal stomach, they have an external stomach where they will exude enzymes in their environment, break their environment down, and then turn that into their body. It's really interesting. It's sort of like a backwards growth of a human. And they do that for who knows why, to grow, to expand, to do all sorts of things. And then those primary metabolites are generally required for something to survive and thrive. So think about your stomach enzymes. Those are pretty important. They're primary metabolites. Metabolites are just another word for compounds that are created. But there's a whole class called secondary metabolites. And all that really means in science is that we don't know what they're there for, but they're not required for the organism to survive. So like they're there, like THC is a great one. The terpenes, terpenoids, maybe they're not required for the organism to thrive and survive, and thrive is a bad word, but to survive and reproduce, but they're there. And then they have secondary effects on other things in their environments, like humans or animals. Fungi are really big about that. So secondary metabolite chemistry is really complex and massive and poorly understood. But on the functional mushroom side, which we'll get into, are some of these classes of compounds. So a lot of the compounds that are found in fungi that we find beneficial for various reasons are called polysaccharides. That's just like an umbrella term. There can be many things that are polysaccharides, poly meaning many, saccharine meaning sugar. So dextrose is a polysaccharide. Sucrose, like these oses are sugars, they're polysaccharides. But fungi are really good at producing many kinds of polysaccharides. Specifically, what we're interested in are these things called beta-glucans. So beta-glucans are these molecular scaffolds. They're massive molecular structures, and they're really complex and have many different attachments on them that do many different things for the human or animal world. And there's not just one beta-glucan. Mushrooms produce many kinds of beta-glucans, but they're interwoven. So this is a cross-section of the fungi cell wall. So fungi, like I said, are made of this thing called chitin. So chitin is this word right here, C-H-I-T-I-N. You might have heard of chitin. It's what crabs are made of. Insect exoskeletons are made of chitin. It's a dense biopolymer, and it's very structurally sound. It's a very sturdy molecular structure. And then interwoven into the chitin layer are a lot of this chemistry in fungi. So this is the importance of why you want to break this down to alleviate those components into whatever you're doing, your broth, your decoction, your extract, or whatever. What that is? Oh, it's the thing on beta-glucans. Beta-glucans are immunomodulators. They modulate the immune system. They're not stimulants, so it's a bidirectional modulation, which means if you have an underactive immune system, it does stimulate it. If you have an overactive immune system, it can ramp it down, which is really cool. So some people's immune systems attack themselves. So beta-glucans can help in that process. Then you have this whole other class of compounds, which is really interesting, called terpenes. This says triterpenoids, triterpenes, but terpenes in general, which you might have heard of from cannabis. They're volatile, aromatic compounds. They're not really heat stable. That's why you can smell cannabis, because they're evaporating due to heat. They're no longer on the plant anymore. So that's why when we talk about low temp extractions, it's really important. Because these triterpenoids can break apart and go away. But they are really cool in that fungi can produce monoterpenes, diterpenes, triterpenes, sesquiterpenes, really complex chemistry. So reishi itself, it's a really cool mushroom. This is Ganoderma lucidum. There's many different Ganodermas. We have Ganoderma applanatum. We have many different Ganodermas here in our backyard. All over the world. Ganoderma can be pretty much anywhere. No one knows what the real Ganoderma is, so if someone tries to tell you this is the real reishi, no one knows. I think reishi is more just Ganoderma lucidum is supposed to be the species binomial, but it doesn't mean anything. Everything was called Ganoderma lucidum. Like, oh, it's a red mushroom conch on the side of a tree, Ganoderma lucidum, Ganoderma lucidum. And then when you run genetic analysis, they're completely different species all over the world. There was a research publication that came out recently where some scientists went to 30-something farms across the world grabbed their Ganoderma lucidum culture, ran genetic analysis on it, and it was 13 different species. Some people say, well, it's Lingzi, Ganoderma lingzi, or it's whatever. But this word Ganoderma lucidum is sort of a placeholder. And then until we run full genomics, we won't really understand the complexity of it. But what turns out, we've grown multiple species of Ganoderma. This one comes from the US. And at least on a beta-glucan side, they're all very similar. So this evolution of the gene clusters that produce the chemistry probably evolved in a common ancestor. So there's probably highly conserved genes there. So as they split into more species, they probably all have the same ability to produce very similar compounds with overlap, but then some novelty too. So there's probably unique chemistry over time as well. I don't know if this is even landing. Maybe. Apologies. So you also have sterols. Ergosterol is a good example. It's the corollary. Can't say that word, but it's synonymous. It's close to cholesterol, but it's a fungal version of it, and it's better for our systems than cholesterol would be. You have statins, which can help also on a cholesterol level. Protein, there's like up to 4% protein in fungi, and there was a study that came out and looked at fungi versus meat eaters, and 8 out of 10 were more likely to be satiated from eating mushrooms than they were from eating meat. It's pretty cool. Immunomodulating, I kind of went into that, but these beta-glucans or these different polysaccharides will lodge themselves in your gut lining, and they'll modulate your entire immune defense system from there. And it can be really complicated. So T cells, macrophages, NK cells, all this stuff is upregulated throughout your gut. And then this term adaptogenic, it's being thrown out like candy, but I want to talk about it in a sense of a scientific perspective. So this word adaptogen came about in World War II. Looking at Siberian tribes, I believe, and interested in night vision, which was really interesting. But this idea of you have soldiers that experience really stressful days and interactions, death, gun fights, whatever, and then they'll have a stress response, like a spike, adrenaline, and then they'll dip. And they're trying to avoid the dip. Like, how can we maintain sort of a stimulated level? That's this word adaptogenic was born. And adaptogens are well known in the scientific sense, specifically for stress. So stress is I'm being vague here on purpose because you experience stress different than I experience stress, but it's still stress, whether it's financial, it's socially constructs like money. It's weird, it's abstract, you're like stressing me out. It could be physical stress, lack of sleep, poor diet, poor exercise, you're injured, all of that stress. So what does that mean in the body? Imbalances, and specifically you can look at hormonal imbalances. So we know the hormone that we're all talking about that has spikes that you'll want to avoid, cortisol, right? So when you take an adaptogen, and anybody know a really potent herbal adaptogen? There's 2 of them that come. Ashwagandha, there's another one, South American herb. It's a warrior herb. Maca. It's a root, yeah. All right, yeah, sorry, not necessarily an herb, but plant. Root, but they're considered adaptogens. So reishi is considered a superior adaptogen. It's ranked above everything else, including ginseng. But those adaptogens will help create more balance in that unbalanced hormonal spikes, if that makes sense. So that translates to better sleep, less anxiety, all sorts of really cool things. But you should obviously focus on that stressor and deal with that. Because there's a lot of cofactors when you're talking about getting benefit from herbs or fungi in general, right? Your diet, exercise, all these things play important roles. Yeah.
So how is it doing that? Because I know there are some genetic conditions that actually prevent you from receiving cortisol at all. So is it functioning on that similar meeting of the receptors, or is it doing it a different way?
It's a great question. I don't know the depths of how that system will work specifically, but it's something that you should let me know when you Google it, because you sound like you know a little bit more than I do about that specific thing. Yeah.
I have 2 quick things. One, thank you for saying that about adaptogens, because I work at a supplement company and we use adaptogens, and there's so many people who call in and are like, just think, like, we always have to be like, you have to also like address the stress. Like, this isn't an antidepressant that's going to like cure your thing if you don't actually like know what's causing you stress or anything. So thanks for saying that. Also, just like, not necessarily relating to this, just everything, because I know a lot of like mycologists and like like everyone's who's obviously taking this class is like, oh, there's like kind of so little known and it's so exciting, like mushrooms are so exciting. And I think there's this general fear in the West of like, we don't know enough and that's scary, and like mushrooms are scary and we should be scared of them. What is your opinion on like where that comes from, and I guess like how to, like, have you seen it in the last decade, like, or so, like, get more normalized and better, and like what is the relationship between?
Yeah, it's a couple things there. I think that historically, through historical use, indigenous use, I think there was a deep connection and understanding with these, and I think that was broken for various reasons. And then you kind of split between the mycophilic and the mycophobic. And the mycophilic, like the national sport for hunting in Czechia, they joke is mushroom hunting. So there's like cultures that are really interwoven into their historical and understanding of fungi and haven't lost that. Then you have more Western societies that kind of took a shift. And then, you know, because of that separation, you have something that's ephemeral. These mushrooms that pop up and go away. Some people are afraid to touch them because they're toxic, and that people get sick and they see die. And they lost that deep-rooted evolutionary understanding of what was edible and what wasn't. And I think that fear plays a big role. A big part is just education, too. And it's like, one thing I like to talk about, too, is that it's not something we're discovering. It's more of a rediscovery. And we're just understanding it through a different lens. But I think education is a huge point, because I still field questions about, will lion's mane make me trip? Like, that's a regular ass question I get every day, you know, and I get emails like, am I going to fail a drug test? You know, these fungi are not illegal on planet Earth. So, I mean, there's just a lot of lack of understanding, and I think that's a big part of it, and I think that's where a lot of the fear comes from, is just, you fear the unknown, and if you don't know enough about it, you're not going to touch it.
I guess it's just interesting because we don't really find that in like herbalism or like as much, I guess.
No, not at all. No. Yeah, most of the...
Like berries and stuff.
Yeah.
Like, aren't scared of eating raspberries or won't, like, do you know what I mean?
Yeah, for sure, because I think it's a little bit more culturally normalized, for sure. Fungi, again, are just this ephemeral organism that kind of comes and goes, and I think that scares people to some extent. I saw another hand. Yeah.
If I'm not derailing or jumping ahead, can you speak a little bit about like the difference between like reishi and, am I saying that right, and cordyceps?
Yeah, I'll talk about them.
Okay.
Yeah, you're good.
Thank you.
Cool. Any other questions, real quick? We're doing great. We got 15 minutes. Let's do this. Lion's mane. Talk a little about Erasium erinaceus. That's the Latin binomial, means hedgehog. I don't know. We call it lion's mane. But there's a few species of lion's mane. We got our hands on Americanum, which grows out in the East Coast through Iowa. We found a culture in Telluride this year, Coralloides, which is also coral tooth, bear's head tooth. I don't know where they get these names. And then there's Erasium erinaceus, which is the classic lion's mane, but they all have common ancestry, and I argue that they all have very similar compound makeup. So there's just not just one lion's mane species, there's actually a couple. When 32 compounds were identified 10 years ago, now we're at 102. That number's going to keep going up, right? Because you don't know, okay, I say this a lot too. See if this lands. I don't remember who did the quote. I never looked it up. I need to figure it out. But you know what you know. Makes sense, right? You know what you know. Sometimes you know what you don't know. You can identify what you don't know. But you don't know what you don't know is infinite. So that alone kind of encompasses the fungi queendom for me, the fungal queendom, the fungal realm. And so what I'm about to say is things that we have understood, but there's so much more to be discovered. So lion's mane to date, paper published 2023, review, specifically identified 102 unique compounds found in this organism. Most of these compounds and the compound diversity found in the fruiting body. The mycelium has, and this is where the mycelium, I don't want you to walk away with this mycelium versus fruit body sort of attitude. I think it's going to be a future of or both together kind of conversation. It's just the product quality is not there for mycelium-based products whatsoever. There's just no evidence. Mycelium itself has evidence purified mycelium is different than mycelium grown on a substrate. And when you cultivate mushrooms, and I maybe cover it, you grow them in a certain process. And one of the processes, as you amplify it from baby to fruition, is grain spawn. You grow it on a hardy grain, and that's its teenage sort of years. It's eating more amino acids, more proteins, more carbohydrates, stuff like that. And then you take that and give it its final adult food, which is generally falls in 2 categories: wood or compost, like straw or something like that, or it's both, like oysters will eat whatever you throw at it. And then it fruits off that. And one thing that's really important on the genetic side is that the genes that are expressed in red are completely different between the body and the fruit. They are completely different. That means that the chemistry is quite different as well. Does that make sense? Morphologically, like when something, like your hand has cells in it, and every cell in your body though, like the cells in my eyes, the cells in my hands, have the same book. But the chapters of which that are red, or sections, are quite different. It'd be really cool if I had eye cells on my hands, but that's not going to happen. It would not be cool the other way around. And that's really important for survival reasons. So fungi are very similar. In the body, genetic parts of the book are red very specifically because it grows through a completely different environment. And the fruit evolved to be very different as well, and it's got quite different chemistry. So there is a really big difference between the two morphologically and chemically and genetically. And when you grow it on a grain, it's highly diluted by the grain that it's grown on. So if we could remove the grain component and create a concentrated mycelium-based product, that's got my vote. We're just not there on a technology scale yet. So the argument between why you would do either or is some people put forth this example. Out of the 102 compounds, many of which are nootropic meaning upregulating the gene expression pathway for neuronal development. Growing neurons is another way to say that. Many compounds are found in the fruit that do this. One that's very, very potent is in the mycelium. Now, when we're talking about consuming it, you need, like, if you get a tiny amount of mycelium, even if it has the most potent compound in it, it's too little to have really a therapeutic value. Now, when we're talking about fruit, when you got a ton of it, and you have multiple compounds in this that have nootropic benefit as well, it's night and day difference in effect. So, and then that argument is spread between all the mushrooms. They're like, well, if I'm doing it for lion's mane, why do they do it in general? So if I broke mushroom cultivation into 4 simple stages, they're not equal. They're not 25% each. Grain spawn is like, getting to grain spawn is like 20% of the work. The other 80% is fruiting the mushroom. So if I could take my 3,000 square foot operation and cut out 80% of the work and sell it for the same price, why wouldn't I? Well, I'm not an evil asshole, so I'm not going to do that. But that's what's happening, unfortunately, because when you read by the way, there's a difference between a nutritional fact label and a supplemental fact label. Quite different regulations, by the way. And unfortunately, in the supplemental world, you're allowed to say mushroom, and people assume that's the fruiting body. So anyway, lion's mane.
So how do you know if it has the fruiting body in it?
You should find a company that's transparent, that says fruiting body only on the product.
So it would actually say that, fruiting body.
If they use the word mushroom, they have really bad marketing team if they are using fruit, because it's well known and you want to promote that stuff. Some companies are also promoting mycelium, so they'll say mycelium. If you see other ingredients and it has a grain, there's your answer. If you're like, it says mushroom, I automatically say just either reach out to them and get clarification, or just avoid the product altogether, because they're hiding behind vagueness right off the bat. What else are they hiding? Can you identify where the mushroom came from? Can you identify the part of the mushroom? Do you even know if it's extracted? Those are 3 simple questions. Reading a label, if you can answer all 3 of those, you're on the right track. I also have a business card right there in front of extracts.
I have a question about
yeah.
grow kits. What do you think about those legitimate ways to cultivate your own mushrooms?
Absolutely. Yeah.
Are some companies better than others, or are grow kits pretty much good in general?
Well, no, that's a loaded question. I can't speak on behalf of all the grow kits. I don't know if one of the best, I don't know if they're a spawn supplier, but Maine Cap and Stem, they're out of Maine. they produce the best quality spawn of anybody in the world. But they might do grow kits. I don't know. If they don't do grow kits, you can at least buy a wood block and just fruit it. It's a grow kit, basically. That's what I would do. Yeah.
Do you find that it's kind of like growing plants indoors in a hydroponic system versus like mushrooms from the wild?
Yeah, another loaded question.
Yeah, right?
Yeah, it's fine. I'll go into it. It's fine. It's a really good question. So I'll repeat it. So in my own rephrasing, if you don't mind. A really good example is sterilized hydroponic cultivation of cannabis versus grown out in full sun, right? Exposure to microbes, exposure to environmental factors, actual sun versus LED lights, stuff like that. With fungi, it's a little more complicated. So you can't always find a solid supply of fungi out in the wild. You don't know the land use history, heavy metal accumulation. You don't know a lot of things about wild harvested mushrooms. Not to say that you should not do that. Like, I go out and find wild mushrooms all the time and I consume them all the time. You just got to be careful. And then the question is like, well, a question would be, are they more potent because of the exposure to the environmental factors versus like a really hyper-controlled indoor environment? Well, you can go both ways with that. So exposure to pathogens and viruses will produce secondary metabolite production, but the energy of what's used is used for various things in the environment where you can control that energy expenditure in an indoor setting. It can also pump towards potency and yield. So I don't think that question's been answered necessarily, but there's reasons you would go either or. Some mushrooms you cannot cultivate either. So you have to wild harvest those. But then when you put this sentence together, wild harvesting by humans, when you have humans in the same sentence as wild harvesting, it equates to unsustainability for the most part, because we don't understand the 30% rule or giving back. We just want to take. So I recommend personally to support local growers in your local environment, mainly because you can have a lot more control over what you're consuming and understand where it came from. And then you can have a way more sustainable kind of approach there as well.
But were there any kind of lab researches into this topic?
No, there's some new stuff coming out. There's a guy on the East Coast. Yeah, it's a neglected mega science.
Okay.
Neglected being the key term there.
Right.
But there's a guy on the East Coast, I forget his name. I met him at the Mushroom Summit in Monterey, California this year in June. And he is working with the university because he's able to he's doing outdoor cultivation and inoculating like elm and a lot of trees out there. And what he's looking at is the microbial communities found on the wild mushrooms. But we don't know what is different between the two yet. We do know that some wild harvested cultivars for food have better flavor because of that complex chemistry of being in the environment, but that could be a created substrate that does that as well indoors. So you can go both ways with it. So it depends on the mushroom, it depends on your goal, it depends on what you're looking at, but I would say right now it's a big question mark, unfortunately.
I have a question that's kind of like stemming off of that a little bit. Do you guys apply any like deliberate stressors to your mycelium to like try to get better yields or try to like get it to produce certain metabolites or something like that? Like I know like with some plants like with cactuses and stuff, you can like stress the crap out of them and get different alkaloids out of them and stuff like that.
Absolutely, yeah. It's a really good.
Apply the same with.
It does, but it is not really being looked at. I think it's a more novel approach to what the future holds for future prospects in mushroom cultivation, which I don't cover necessarily, but it's a good topic. One thing I'm very interested in is called elicitation. So elicitation, so when you stress an organism out, it can upregulate the secondary metabolite biosynthetic pathways. It can produce more and more compounds. Targeting that is really complex, so there's no way to know, am I going to have more neutropicness out of this by stressing it out from heat, from water withholding, or when there's an insect outbreak. What you can do, though, in a very controlled way, instead of releasing a bunch of insects in your grow room, there is a hormone that is produced that is produced by the organism that triggers a lot of this, right? So you can actually get the hormones, and you can treat your grow room with that, and it's like it's being attacked by insects, and then you can measure the outputs of that. None of that is being looked at. Yeah. It's a novel future prospects sort of thing that I'm actually curious about as well. but there has not been enough information on it.
So basically, we're saying that we're really very ignorant about mushrooms for now.
Oh yeah. So the iceberg analogy is like way off. Like imagine like, I don't know what's under the water, but it's big. Yeah. Yeah, if we were building like, I don't know, the Empire State Building, like we have a brick. It's probably a good analogy. Any other questions? These are great questions, though. All right, lion's mane. Yeah, you know, what's really cool, lion's mane's got many different compounds in it. We use it with I put the blinders on for people because or else it results in analysis paralysis. Mushrooms can do so much. We know very little, but more and more is coming out, and they're more and more like, we had no idea. It's not just about neutropicness of lion's mane. They also have beta-glucan, so there's an immunomodulation factor. They have really cool studies on, they do a lot of benchtop studies, and one of the things they test against is HeLa cells. Are you guys familiar with Henrietta Lacks? Also a great podcast. I think Radiolab did one on it. It's pretty cool, but they biopsied one of her cancer cells. They took some of her cancer cells in the 50s, and it was an immortal cell line. And that doesn't mean like you can't kill it. It just means it keeps reproducing without aging. It just and they have it today. And what they'll do is they'll test many things as a benchtop study. Will it kill HeLa cells? And all the mushrooms that I'm going to cover here do, which is really cool. But these are benchtop studies, right? They're very controlled, petri dish or test tube type studies. Way different than human clinical trials, but they're the first step in getting to something like that.
Here's a question.
Yeah.
You said that mushrooms fight the same things that we fight. Are you saying they fight like disease and things like that?
Yeah, exactly. Yeah, and we're closely related. So, and they have the ability to produce the chemistry to fight it, and that's why when we consume it, we get that benefit as well. If that makes sense. Oh yeah, this was pretty interesting. Between 2002 and 2012, so 10 years, over 244 drugs were tested in 314 clinical trials for Alzheimer's disease, but only one was approved, and that one was not proven successful. Harvard Medical Journal, I pulled this from Robert Rogers. If you don't know him, write that name down, Robert Rogers. He wrote The Fungal Pharmacy, 2015-ish. It's a reference text. He personally had a lot of it translated because a lot of the research is not done in the US because of our privatized system, and we don't have to go into that necessarily, but it's not conducive for figuring this stuff out. Most other countries are doing it. But it's got over 1,200 citations, and it covers a whole gambit of wild harvested or functional mushrooms. It's a big reference text. He came out with a smaller, concise text called Medicinal Mushrooms, the Human Clinical Trials. It's a lot smaller. Came out in like right after COVID. It's a really good book. I reference a lot of it. This came from there. Another one came from him right in the introduction. And I source checked him because I was like, well, this is crazy. Harvard, and it straight out of Harvard Medical Journal. If you were to sum up this cause of death for the last 2 decades, it would be the 4th leading cause of death in the United States. And this cause of death is taking prescription medications as prescribed by your physician. Let that sink in a little bit. I said that at a pharmacy summit they invited me to. And it was well received. I was scared to say it, and I was like, I'm going to do this in a room full of pharmacists.
Like we all knew.
Yeah, they're like, yeah, tell me something I don't know. I'm like, oh, God.
Well, how many times did you speak at that conference?
Yeah, it's true. But these were mom and pop pharmacies too, and they're yeah, I won't go into that. It's really interesting. They consider themselves PhD glorified pill counters, and they want to have more control over the old school pharmacy kind of approach. So they're really expanding out to over-the-counter options for people to help target what they're working with. Your entire, we're going to lion's mane, but it's not just about your brain. We're talking about neurons. Your entire body's made up of neurons, it turns out. Lion's mane is really good at repairing damaged, broken, not connected, or whatever. Neuronal damage even in your brain. It's neuroplasticity. You have changes all the time based on learning. Lion's mane is really conducive to helping with these connected pathways. And they connect through to your heart and your lungs. They connect through these myelin sheaths, and then you can have this degradation of myelin sheath occur for various reasons, or amyloid plaques, that's Alzheimer's, all these different things. And then lion's mane is being looked at to help repair these and create new connections and just a lot of cool stuff. Then you have the Ganoderma complex, the species complex. Again, there's not just one reishi. I call pretty much any reishi that we grow. We have like 12 or 14 species of reishi, different Ganoderma species, and they all, so far, have really good functionality for the human body. There's a lot of historical context with reishi in Asia. And again, it overlaps a lot with Egyptians in that it was not allowed for commoners to consume these. And if they were found, they should be on their way to the palace to give it to royalty, or they'd be executed, like if you were caught with these mushrooms. A couple pictures of different kinds of reishi. I think antler reishi is one of my favorite aesthetically looking reishis. It looks like a coral type mushroom, but it grows these intricate antler patterns. Ganoderma multipilum, multiple pillar in Latin. And then you have your quintessential red reishi, which is, whatever. Some have these. This one's a white banded one from the US. We have cordyceps. Cordyceps militaris specifically, the alien Cheeto, if you will. But cordyceps is pretty cool. So, and I won't get too much of the backstory, but historically it was consumed for a very long time. And what the cordyceps genus themselves have, who knows how many different species? Over 700 in just South America have been identified. Each insect generally has a cordyceps that'll infect it. It's considered an entomopathogenic fungi, a parasitic mushroom. But when in the world of ecology, when we hear parasite, the common person who's not familiar with ecology thinks negatively. But I want to redefine that, because if you have a complex food web or interactive biological system, when you measure that, it's called biodiversity. And biodiversity is the measure of different unique genetics in an ecosystem. The more biodiverse a system is, the better it is. What does better it is? I mean, various different things. Be able to rebound due to stress, environmental disaster, everything. So a non-biodiverse system, as comparison, would be a monoculture, which would be like a cornfield. That's why you have to apply heavy pesticides, heavy herbicides. If they're genetically identical, it's the scariest thing that you could have, because anything could take it and wipe it out. When you have that biodiversity, it allows a lot of complexity for survivability. And when you have something like an insect in that ecosystem start to dominate and take over or invade various reasons, it starts to drive down biodiversity. Not a good thing, right? So a species like cordyceps will come in from a top-down control and infect it and drop its numbers to a manageable level, which and then raises biodiversity again. Does that make sense? So this trophic cascade thing. So parasites equal good.
So I have a question about cordyceps.
Yeah.
So like in Chinese medicine, it's the one that actually has the worm.
Yeah, I'll get to that. Yeah.
Okay.
Cordyceps senescens.
But
So
I want to know the the medicinal or functional differences between the non-worm one
Yeah.
versus the worm one.
yeah, we'll get there. Perfect. Great. Preemptive. Keep me on track. So historically, Cordyceps senescens was the consumed mushroom. It's not cultivatable. It's only wild harvested. Again, wild harvested, humans, and sustainability. It's found in the Himalayas at a certain elevation at a certain time of the year. It's currently worth more than its dry weight in gold. It's a very expensive mushroom. And it became reclassified as Ophiocordyceps. And various things kind of went into that. And it's known to, its energetics is what we'll get into, what it's known for in the energy side. But Ophiocordyceps, you can't cultivate, and it's a compound species, it turns out. It's sort of like a lichen. So a lichen is does anybody know what a lichen is?
Yeah, but we want you to describe that.
It's a compound species made up of 3 different things. A fungi, a yeast, and an algal cell. And then if you take them apart, they don't come back together, unfortunately. And you can't take those 3 and make one. But they co-evolved into this complex compound species. So Cordyceps senescens is also a compound species. So the compounds we're interested in, which I'll get to, are only found in the wild harvested version because it's a complex arms race between the 3 species as it infects the caterpillar. And then it produces the compounds in the fruit. So a lot of times when people clone this, they get one of the 3. and then they'll grow it out on grain, and then they'll sell it as the actual species, and it has none of the benefits because it's not the compound species. But there is a mushroom that's related called Cordyceps militaris, and it's cheaper than gold to grow. And because of breeding techniques from underground breeders, it has higher concentrations of those beneficial compounds we're interested in than the wild harvested version. Higher.
Wow.
Higher. And those compounds we're interested in, specifically, cordycepin is the most famous one. So if I did my present, howdy dog. I don't know who you are, but you're cool. So cordycepin, they name the compounds after the Latin binomial, usually cordyceps, cordycepin, whatever. Cordycepin. So I venture to guess there's at least a couple nerds in here. Help me out here. The mitochondria is, thank you, the powerhouse of the cell. I was a high school teacher. I understand. I was also in high school once. So you were force-fed that, and you were forced to memorize that the mitochondria is the powerhouse of the cell. Let's put some context to that. Let's make it make sense now. Instead of memorizing, you're going to have some more tools to understand what that means. So the mitochondria is found in every cell of your body. It's your energy currency. It utilizes certain aspects, amino acids, to create energy called ATP. So I'll come back a little bit. Adenosine triphosphate. There's a whole pathway in there. It uses adenosine and creates this energy. And that's why I'm moving right now. Without this entire thing, you would die within not even any time because you couldn't respire, you couldn't do anything. You would just not exist. So the mitochondria, ATP. So adenosine is what's important here. So if I did this right and I had a compound, if I had adenosine on the left here, and then I had cordycepin here, you can see one difference. This OH group. They're so similar that your body doesn't notice. So when you put cordycepin in your system, you can upregulate your metabolic pathways, and that can translate to various different things. It can be, in general, lung capacity. The way I like to explain it in its simple terms is I hike, I bike, I run. I did jiu-jitsu one time. I'm kidding, for like a year. But you get winded. Jiu-jitsu for sure. Like you get rolling on a mat with somebody, you're like dead. Or hike, bike, run, or whatever. You hit a wind wall, is what I like to say. You get winded. So taking cordyceps for a prolonged amount of time can move your wind wall incrementally for you, so you get more output. And that's through the metabolic upregulation system. It's pretty cool. But yeah, your question.
So there's, I hate to say a ton of research, but there is like significant-ish research on lion's mane and dementia. What was it? Alzheimer's?
Alzheimer's and dementia, cognitive decline in general.
Are they doing the same for cordyceps and
Athletes.
functioning?
Yes. Yeah, so they're starting to do a little bit more. There's some information come out with COVID, which is cool because there's a ton of like antiviral stuff going on here. But yes, I mean what kind of propelled this in the 90s, the women's Chinese athletic Olympic team won like 8 gold medals, and they're like clear doping. Like they weren't. They're like no, they were taking cordyceps every day. And the thing is though, is high Olympic athletes are not getting that much benefit. Like it's like shaving your legs if you're a pro swimmer, right? Or biker or whatever, shaving your, sure, fractions of a second. I think the people like myself who are on their feet 12, 14 hours a day, working, the working class, I think they get the most benefit out of something like this. Co-factors included, right? Diet is a big part, exercise is a big part, but yeah.
I work with a lot of kids, so all the mushrooms that you've talked about so far, are they kid-friendly and safe? Is there like an
Ask me off camera. That's why I gotta be careful about talking about some things, referencing in business. But
Are you gonna say yes or no off camera? Stay tuned.
no, I'm just kidding. There hasn't been any clinical trials with kids. I think there's probably some ethical things going on there. But fungi are a food group.
Because in the herbalism world, there's like very definitive answers. And
Right, like echinacea might not be the best all the time, every day for a kid or humans in general. It might be needed as a stimulant over that kind of stuff, for sure. I think there are probably very similar ways to navigate fungi. But the way I would look at it is if it's in the food side, because you can get into medicinal functional and not edible. And then you can get into the edibility side. So lion's mane falls in both. I have a 2-year-old at home. I feed him lion's mane all the time. We make non-alcohol extracts, glycerin versions. I give him everything but cordyceps. I don't think my little kid who's already off the charts in energy needs more.
That's interesting because like my daughter has like functional like sports-induced asthma, so that's what kind of triggered that. Because they already do a lot of things for like biodiversity. But
Yeah.
would that be helpful?
Yeah, it's just the cordycepin side, I avoid that with my kid because I was like, I think his metabolism's fine. But he loves the reishi and the lion's mane, flavor-wise, for sure. Yeah. Any other questions? Cool.
I have just a comment on that. I think we don't give kids enough credit of like they know what they need, their bodies kind of. I know you were talking about energetically. I feel like a lot of kids would have that very innate. They're like, I don't like this, and there's a reason.
Yeah, that's true. I can agree with that. But there's tons of complex chemistry going on in cordyceps. They do all sorts of cool things, and I won't go crazy into cordyceps, but it's one of my favorite mushrooms to consume.
I'm so sorry, just to conclude on that. So we're saying the non-worm one is actually better.
If you have the right genetics
Yeah.
and the right extraction.
The ones you grow, you're happy with them, right?
I'm happy with the genetic. Yeah, we don't. So cordyceps is an interesting fungi in general. Cordyceps militaris, and I don't know if this is a blanket statement for all of them, but it senesces. It dies after 6 months. I have clones of lion's mane. I've kept alive for 10 or more years. Cordyceps will stop fruiting after 6 months. It's got some sort of clock on it genetically, and it senesces. And we haven't cracked the mycological cultivation community hasn't cracked the code on removing senescence. So you have to rebreed it every 6 months. It's a full-time job. We don't do that.
So
We grow them, but we don't breed them. We source genetics from a very specific breeder who's been doing this for as long as I've been cultivating.
Do they breed them with the worms?
No, Cordyceps militaris specifically. Cordyceps militaris is a species I'd recommend consuming. And then as long as it's a fruiting body product, it's probably there's only a handful of breeders in the United States. There are Cordyceps militaris outside of the country. They're not very potent. They're just not. I've run tests on them. They're just not great. But the stuff that we grow from the genetics we source are insanely high in numbers.
Has every batch?
It depends on what you're doing on the extraction side, but you do batch testing, yes. And we don't test you can't test for, so.
Every 6 months?
Yeah, sure.
Consistent?
It depends on the genetics, because this guy's figured out how to make albino versions of it and variegated versions of it, and they're slightly different, but the beta-glucan values are very similar, and the cordycepin goes up in the albinos for some reason. But you can't test for every compound. There's no economical way to test quantify percents of everything that's in a mushroom, because the way you test for that is having a reference standard, and there's not the one reference standard for lion's mane for erinacines and heracinones is like $25,000. So these labs they can't do it. Like, okay, yeah, you want to do a lab test? $25K. Cool, sure. All we can test for right now are beta-glucans. And that's really why you test for that one, is because it's found in pretty much all functional mushrooms, and it's a quality control, but it's not used as an indicator for a better product. It's used as an indicator for knowing if it's fruit body versus mycelium. Because if you'll breed a industry of isolations if you're looking at just beta-glucans. Beta-glucans are immunomodulators. If I want the neotropicness of lion's mane, I don't give two shits about beta-glucans. But I would like to have a beta-glucan reference to understand, is this fruiting body or mycelium? That kind of is used as that tool. Does that make sense? So to run a full lab analysis and quantify the percents of everything in cordyceps, one, you can't because you don't have all the reference standards, and two, it would be insanely expensive. So this will grow over time, and I refer to it as the potency problem. We can talk a bit about that in a little bit because I'll talk too much about it, but good questions. Turkey tail. If there is land mass and wood, you'll find turkey tail. It's one of the widely distributed mushrooms on the planet. It's found in every continent except Antarctica. And again, if there was wood and the growing temperatures, it would be there. Turkey tail, yeah. Trametes versicolor. We'll just kind of briefly go over this guy, but it's really well known for gut health. It's a prebiotic for the gut, and it's a really potent immunomodulator. It's really big on these polysaccharides, these beta-glucans. Specifically, two beta-glucans that it produces that are really famous are PSK and PSP, polysaccharide crestin and polysaccharide peptide. But these guys are powerhouses when it comes to immune support. So I think about turkey tail as an immune support kind of mushroom. And then there's really cool stuff coming out. One study I like to cherry pick specifically was a clinical research trial on cancer patients, 186 patients, I think, and they split them into two. You have a control group. They're both taking chemo because you can't withhold chemo from a test group. It's unethical. And again, I am not saying to not do chemo. That is not what I'm about to say. There has never been any evidence or any support of withholding modern scientific treatment protocols and having success. Zero. Is that clear? Because what I don't want someone to walk away here and tell other people is take turkey tail, it'll cure cancer. No. Nobody said that. In conjunction with chemotherapeutics in this study, there was a control group just taking chemo, and there was a test group taking chemo plus turkey tail extract. The turkey tail extract group outlived the chemo-only group by 400%. And it has something to do probably with turkey tail's ability to protect healthy cells from free radical radiation. When you have chemotherapy, its goal is to kill anything, healthy cells, cancer cells. For some reason, when you have turkey tail in your system, it protects the healthy cells, but doesn't protect the cancer cells. And they're still trying to unpack a lot of this stuff. But again, on the functional mushroom side, if you were to sum up the clinical research or whatever across everything I just talked about, and I had to theme it for you and just summarize it, preventative maintenance is the goal. If you're looking to treat, too late. It's just, maybe it will help, maybe it won't, but most of the research is on preventative maintenance. So it's good to start now, today, than to treat anything. Does that make sense? So taking a functional mushroom for its functional benefit based on clinical research, you have to take it daily, hopefully a therapeutic dose, and I'm not going to define a therapeutic dose because genetics are variable, extracts are variable, no one really knows what exact dose is. So a therapeutic dose on a daily basis. When do I take it? Doesn't matter, just take it. And then the last one is for a prolonged amount of time.
But should you cycle through like a lot of herbs you're not supposed to take?
Functional mushrooms in general,
Yeah.
to get the benefit, you take them daily
Okay.
for a prolonged amount of time and at a therapeutic level. I know what you mean, like echinacea is a good example. That's still contested
Like
too.
horsetail, like take it for 6 weeks and then stop.
Right. With functional mushrooms, when you're looking at the, well, I'm basing this on clinical research specifically. There hasn't been any sort of evidence like that yet. I'm sure there might be like a very specific protocol when to take it, when not to take it. But the clinical research is done for a very long amount of time. Like we're talking, they'll take samples like 3 months in, 6 months in, 18 months in, and they'll just see better and better results in general. Yeah.
So, kind of piggybacking off of that is, you said like you don't like dosage is variable.
Yeah.
So, is your opinion kind of like, and I know this is not the actual how dosage is, it's not like grams or whatever, but would it be like, well, 1 gram, like 5 grams may be more powerful than 1 gram, but 1 gram is better than 0 type thing? Like a little bit is better than.
And there hasn't been any dose-dependent response studies, so there's no understanding. There has been one.
Mushrooms versus taking the tincture. Like if I just eat turkey tail every day versus taking a
Yeah, good luck with that.
tincture.
Here. So turkey tail is a lot like that. Take a chunk out of that right now. I'm just kidding. But it's not an edibility. You can't digest that. It's like balsa wood or corkboard. But you can treat it in a way to make it digestible by breaking the chitin down through extraction. So I would recommend, or you can like a decoction kind of thing, right?
You should focus more like instead of eating lion's mane,
And that,
trying to, go to the farmer's market and have that be like part of your diet. Just usually the medicinal tinctures you get more value.
so that's a relative thing, right? So that's why when I was talking about you can have that. When I was talking about my spectrum, do you want it? I was going to give everybody one, by the way. So, yeah. Thanks for coming. You get gifts. So on that spectrum, my measurement was for functional benefit, right? So if I'm hyper-focused on that, for functionality, extracts are the way to go. For health in general, eat mushrooms. Eat them. Like lion's mane's great. Lion's mane steaks, lion's mane tacos. If you have an air fryer, foolproof way of all sorts of stuff. I eat mushrooms and I also take extracts. I'd say do both because it's really good protein and various things for other parts of your body by eating the actual mushroom versus an extract. You get benefits both ways. But if you're looking for the nootropicness of lion's mane, you want to have a better controlled product because when you cook it, are you controlling for temperature? How long are you cooking it? Like I don't know what kind of stove you have. Like did you use oil? Did you not? So if you're looking for very targeted therapeutics, extracts is the way to go. Yeah.
Are there any known side effects, interactions?
Alcohol. Yeah, there's been some stuff coming out lately with reishi and alcohol. And anybody know inky cap mushrooms? So you guys know the pharmaceutical drug that's given to alcoholics to prevent them from drinking. It makes them violently ill. I think something like that. Yeah. So that the effect you get the hangover, it basically accelerates that and makes it immediately. And it turns out that inky caps does that exact thing. If you eat inky cap mushrooms and drink alcohol, you'll get violently ill. There are side effects. They're interesting, but few and far between. So like reishi apparently has been known to naturally be a blood thinner. So if you are on blood thinners, it's good to talk to your medical professional before you start a regimen on a functional mushroom. Unfortunately though, just a quick anecdote. So when my partner was pregnant with our child, we went to, what do they call the baby doctors? Not the baby doctors, but when you're pregnant and you go to the doctor. Maybe, one of those things. Super awesome doctor, but she oversaw my wife during her pregnancy. And I asked her, I was like, is it safe for my wife to take my extracts? I make them, and I haven't seen anything, there's, maybe I wouldn't give her cordyceps. I really just want to give her reishi, because when in doubt, reishi, that's what I say. It's the most published clinical research mushroom on the planet, along with turkey tails getting up there. And she told me, honestly, I'll defer to you. She's like, because I don't know enough about it, I have to say no. But because you know more about it than me, I would ask you. And I was like, interesting.
So did you test it out?
Yeah, she definitely took lion's mane and reishi for sure. My kid's great.
No, but what about her?
She takes them.
Did it help with, the symptoms?
I don't know. I don't have a comparison. So.
You have to have another child.
Yeah, I'd have to have another child to compare. Do a control. Yeah. There is something known as reishi babies. You can look it up. It's an interesting thing. A lot of pregnancies and a lot of women do focus on reishi as well as give it to their children. It's really interesting. But yeah. Yeah, and we'll just dive into. Oh, okay. We're gonna skip ahead. We got 10 minutes. There's different kinds of mushrooms, different categories you can talk about. In the ecological sense, this is more the ecological sense. There's mycorrhizal. They associate with different organisms. They can produce mushrooms or not. Ectomycorrhizals are like your chanterelles, your boletes, your amanitas. They associate with a symbiotic relationship with some other plant, and they need that to be able to produce the mushroom. So they're mostly wild harvested. They're not cultivated. Morels is another example. There's the endo versus ecto, and it's just how they enter the actual cells of the plant itself, and they can be connected to hundreds of other plants in that system. If you're interested in sort of the research in forest ecology and understanding the fungi beneath our feet, Dr. Susan Simard did a lot of the pioneering research. She got a paper published in Nature in 1999, and she was definitely given a lot of stress because of the hierarchy and patriarchy of male science domination. But Susan Simard, Simard probably, Finding the Mother Tree, can't recommend it enough. Great book. Endophytic fungi, it turns out we have not only plants but us, we have a microbiome, we also have a mycobiome externally and internally. It's really complex. Plants do as well. It helps with their immune activation, their SAR, all these really cool things. And they have epiphytic fungi that live on the surface, and they have endophytic fungi that live inside of them. And one thing that's really cool about endophytes is that they are being discovered to be what's producing some of the secondary metabolites found in plants. It's not even the plant itself, it's an endophytic fungi found within the plant, which is really cool. And then if you took the genetic code of some of these endophytes, they're massive. And they turn on and off sequences based on environmental factors. It's called epigenetics. And if you took all of those silenced genes and you kind of calculated the amount of natural products that could be created, this is the number. So there is a future of discovery of natural, it's called nutraceuticals, of natural medicines through plants and fungi interactions. Cordyceps senescens, cordyceps students thing. There's other cool stuff in here. My hands are so cold. Saprophytic fungi, they recycle things back into our ecosystem. They break complexity down into elemental parts. They do this through this key and lock sort of analogy. Enzymes break things down to simpler parts. Fungi are really good at this, which can get into microremediation. And microremediation is something that I'm super passionate about. I think it's a lot of fun. It's this applied mycology sort of side. And you can do so many different things, and it's more of a complex interaction between not only just fungi, but plants and microbes to be able to really clean up an environment. And I worked in this field, maybe called probiotic agriculture, probiotic farming. I was a professional microbe herder for years for soil science and cannabis cultivation. And you focus on all of these different parts to have like a really complex interaction of breaking complexity down to simple elemental parts. And fungi are really cool in that they can break complex hydrocarbons, petrochemicals, PAHs, pesticides, forever chemicals, glyphosate, you name it, into CO2 and water. So so if you looked at the molecular structure of wood, otherwise known as lignin, it's a complex hydrocarbon bonded molecular structure. So it's not like they're just like doing something new. They understand how to break hydrocarbons down. And if you can train a fungi to solely persist on these complex chemicals and utilize it as food over time by training the fungus to do this. And this is more of a more modern sort of approach, but you can use it as a mycofiltration, what I kind of talked about, cleaning up waterways. I'm hopefully working on a project in Bear Creek with E. coli. There's a big project through MycoReach through one of the founders, Jeff Ravage. He did the Cold Fire Project. And it's literally, so a lot of people I want to educate about microremediation is because they read something cool and then they try to do it at their environment. And they're taking this random mushroom from who knows where and applying it and failing. So the complexity of microremediation is you have to do a site analysis. You have to go to the site, understand the contaminant, find what's already growing there, and then what is it growing on. And then you have to run benchtop studies to find out, is it already adapted to breaking this down? And then you have to naturalize it on the environmental wood, if you will, amplify it, and you can re-inoculate that environment with it and amplify it, and it can help break down the chemical pollutant in that environment in a crude, quick way. And that's really what MycoReach is kind of focused on too. Current and future prospects. This is a slime mold example. I always bring it up. I always show everyone because everyone's heard of it. But what they did is they took the Tokyo railway system in the major cities, and they put oat food as the major points, and they let the slime mold grow out, and they watched how it would collapse and then create highways. And then they used that to compare to the current railway system, and it was better. So you can use these organisms. This is a slime mold, so it's a little bit different than what we've been talking about. But they're very, very good at figuring out complex puzzles. And you can use them in an engineering sense to figure out really complex puzzles as well. And then even for food, so this is soy, this is chicken, this is a high resonance microscope. What are they called? Whatever, like an electron microscope. And then mycoprotein. It's blowing up right now because of its ability to create really cool, nutritious, and healthy alternatives to meat. Corn, Quorn, is a UK business that's been doing this. There's one that's just north of North Glen, Thornton area, Meaty. I think they're out there. MycoWorks. They're more of a material base. That's what we're looking at here, like myco bricks, like growing mycelium in the shape of certain things and utilizing it for packaging materials, clothing, myco leather. The new electric Cadillac that came out, I think it was MycoWorks, created the leather seating out of reishi for it, which is really cool. This is just a 3D printed chair made from mycelium in 3D printing. There's really cool stuff that's... These people made them, but this is like something that you'll probably have access to at some point in the future. But this all kind of comes down to the future of circular economy, and I think that's what brought me into the fungal realm, and that's kind of what I started with, is my little backstory about closing loops in our society. If you think about just like mushroom farming itself, you can take things that are waste from the environment, from breweries or wood pulp from paper mills or whatever. You can grow fungi on them, create food or medicine for people, and then what you're left over, the spent material, is compost that can be given to farmers to be inoculated as a compost for their fields or whatever, and it can actually enhance the health of that system in general for various reasons that we've covered. And that's a quick example of a loop there, where you're taking a waste stream, turning it into a product, and then your waste is not waste anymore. It's actually implemented back into the environment. And there's many ways that this can occur and will occur as long as we educate people about these topics. One cool thing about designer pharmaceuticals, it's going to revolutionize the pharmaceutical industry. If Trad Cotter, he does Mushroom Mountain on the East Coast, if he disappears, it's the pharmaceutical industry. What he's been experimenting with in ruffling feathers is, imagine if you could take, someone has a bacterial or whatever, viral infection or whatever, take a mouth swab, put that mouth swab on a petri dish, put like a matsutake or a maitake or some sort of mushroom on that dish, let it produce chemistry to fight whatever that mouth swab has, and then you could take that as a personalized pharmaceutical to fight off what you're doing. Like, antibiotic resistance is real. It takes 10 years to develop a new antibiotic, and it's accelerating. It takes the same amount of time to create a new compound to fight antibiotics and to fight bacteria, but it takes the bacteria shorter and shorter amount of times to figure out how to get past it. So we're going to hit a spot where MRSA is a really rampant issue, and I think this is sort of a solution to that problem. There was a paper that just came out like 2 weeks ago where they took king oyster mushrooms and they took the mycelium and they connected it to a computerized system where it could have control over motors and move, and they would shine a light on it, and it allowed it to control. It wasn't really targeted or directed. It's really kind of weird and simple, but based on the way they built it, it was able to pick its legs up and move based on light energy, which is like insane. I don't know what biocomputers, that's what's coming out next. Perfect. That was kind of a, period, done. Well, questions, if anybody has questions. Yeah.
So I saw a presentation by Trad Cotter like 4 years ago.
Yeah.
And I was curious because he was talking about that. Like, he was like, we just took a block of mycelium of oyster mushroom, injected it with some bacteria culture from someone's like mouth, like a swab or something.
10 days, because he was doing some testing at the time. 10 days is how long it took for the antibiotic.
And then he was creating the metabolites, right?
Mm-hmm.
Are those just something that you can just take that liquid that comes off of the block and consume that, or is it something more like you have to separate the functional part of it, and then administer that to a patient?
I don't think there's an answer to that question. It's a very good question because it could be producing multiple compounds, but I'm sure how that research would probably go is some way to use like NMR or complex laboratory equipment to identify the molecular structure or structures, and then figure out how you can either amplify it in an organic sense through utilizing the mycelium, through biofermentation probably, like liquid culture broth and stuff like that, or sort of like 3D printing the compound if you know the molecular structure and you can synthetically create it and create it in a certain way. But there's a lot of like dose dependent stuff, and there's a lot of stuff to still needs to be figured out till we get to that point. But he's been working on it for a minute now, and I think hopefully we'll get to somewhere where there's some research on it in the near future, I hope. Well, thank you. Hopefully people learned something today. Definitely take a mushroom on your way out.